In the previous video, we discussed about NEET in a detailed manner. We will provide you a link in the description. Now here we have very important topic for discussion. This is known as chromosomal mutations. My dear friends, these are the changes which occurs in normal chromosome, right? And these changes or mutations have broadly two types. Number one is numerical change, and second, very important, this is known as morphological chromosomal mutations. Now, as the word indicates, numerical changes are those which affect number of chromosomes. Means either addition or loss of chromosome takes place in the body is a numerical change. Now, morphological change affect morphology, structure, arrangement of genes of chromosome. Later, this numerical change has two subtypes, euploidy and aneuploidy. To watch an euploidy in a detailed manner, again I am repeating, we are going to provide you a link in the description. Here let us talk about euploidy in a detail. Now what is euploidy? Euploidy means addition or loss of chromosome set from genome of an organism. Means we are talking about chromosome set. Mind this word. Suppose we have this chromosome set in an organism. When one more set gets added, this is known as euploid, and the phenomenon is known as euploidy. Now, according to aneuploidy here, not set but one or more chromosome gets add or delete. Right? Suppose this is set of chromosome in an organism. Here one more chromosome gets added. So this is the case of aneuploidy. Hope you got the concept. Now coming to the main topic of chromosomal mutation, that is euploidy. It will be first video of chromosomal mutation. Now what is euploidy? We have already discussed. Here set of chromosome gets added or lost from an organism. It has broadly two types, monoploidy and polyploidy. In monoploidy, loss of one set takes place. Polyploidy, addition of chromosome set takes place. Let us define. When one set gets lost from diploids, it will form monoploids. It is denoted by one N, simply. Now we have one more important term. Never get confused here. What are haploidy or haploids? When the organism has half set of chromosome in a body, in a cell, it is known as haploids. Let me explain it in a better way by this example. We have plant wheat. It is hexaploid. Chromosome number 42. Each set has 7 chromosomes. Right? Now, what will be the monoploid of wheat? We can simply write monoploid of wheat contain 1n single set that is equivalent to 7. Okay? Now, what will be the haploid of wheat? We can write haploid wheat has half of chromosome, half of 6n is 3n, equivalent to 21. Right? So here monoploid is 1n, haploid is 3n in the wheat. Now one more important point here. Let me write. In diploids, suppose humans, we are diploids normally. We contain two types of chromosome sets, 23 plus 23. Right? Let's assume one set gets lost from a body. When in diploids, when one set gets lost, it will form haploid as well as monoploid. So in other words, we can say in diploids, monoploid and haploid is same. Hope you got the concept. Now jumping to the very important part, that is polyploids. Second part of euploidy. Polyploids means addition of set of chromosome takes place in genome of an organism. Suppose we have organism, this is diploid. Now we can add one more set of chromosome, it will form triploid. We can add one more set, it will form tetraploid. Accordingly, pentaploid. We can write hexaploid, or we can give name simply, these are known as poly, means many, polyploids. Right? Now on the basis of what type of set will get to the genome, polyploids have two types. Number one is known as autopolyploid, same set. Auto means same. Now here, second one is allopolyploid, when different set get added to the genome. In autopolyploid, addition of same set of chromosome gets added to genome. Suppose we have this chromosome set. Again, we can add same set in the species. We can add again same set. So this is the case of autopolyploid. For example, we have potato. It has near about 4 same sets. So it is autotetraploid with chromosome number 48. Right? Accordingly, we have one more plant, banana. It has set of chromosome 3 times, same set of chromosome. So it is triploid. Simply with chromosome number 33 having 11 in each set. Now what is allopolyploid? We can write addition of different set of chromosomes to the genome, different set. Suppose this is normal, we can add different set, again one more set to the genome, it will become allopolyploid. For example, bread wheat. Normal wheat is allopolyploid. It is 6N allohexaploid. 6 different set of chromosome it possesses. Right? Now we are going to add very important and significant topic in this lecture. This is origin of wheat, or we can say evolution of wheat. You know, normal wheat, or we can say bread wheat, it is also known as Triticum aestivum. Now this is allohexaploid wheat having chromosome number 42. Each set has 7 chromosomes present in it. How this wheat has been originated? Let us discuss the story. In the world, there was wild type of wheat species, namely Triticum botaicum. This species is also known as Triticum aeuglopoides. Now by natural selection, through natural selection, this species has been changed into very important type of wheat called Triticum monococcum. It was diploid in nature, having chromosome number, we can write here 14, 7 plus 7. Now after the formation of Triticum monococcum, this species fuses with unknown species of wheat. Some workers consider this unknown species Aeuglops speltoides. It is also diploid species with different genome. Right? Now fusion of Triticum monococcum and Aeuglops speltoides, they interbreed and they form a different types of wheat species, or we can say F1 hybrid wheat. But this wheat do not produce viable result. Right? This F1 hybrid wheat was also diploid in nature. Then what happens? Chromosome doubling takes place in F1 hybrid wheat, and it results into the formation of very important type of species Triticum turgidum with genome capital A, capital A, capital B, capital B, and we can say equivalent to 4N. It was tetraploid in nature. It is also known as Triticum durum. It was the species that has been cultivated for years. Now after their cultivation, the species has been also crossed with one more wheat species Triticum torchi with different genome. Now after interbreeding, they again form what we called it as hybrid wheat. Now this hybrid wheat is basically triploid. It got half of chromosome from Triticum turgidum and Triticum torchi. So it is a triploid, but it was non-viable. It do not produce fertile seeds. Then after chromosome doubling of this wheat, it formed normal type of bread wheat called Triticum aestivum with 6 set of chromosomes equivalent to 42 chromosomes. So this is case of allohexaploid. Dear friends, it was all about origin of bread wheat. I hope you got the concept by all means. Now if you find this video valuable, kindly share, never forget to subscribe. Thanks, have a nice day.